Good day, this is Professor Will, CBMD PhD. Today's Thursday, March 25th, 2021. It is 7:17 and 7 seconds p.m. Eastern Standard Time. Today's research, brain barrier. So let's hop to it here. Leaky blood-brain barrier linked to brain tissue damage and brain aging disease. Date, March 24, 2021. Source, American Academy of Neurology. Summary. Now, a new study has found that people with cerebral small vessel disease, who have blood-brain barrier leakage, had more brain tissue damage over 2 years than people with less blood-brain barrier leakage. As people age, changes in the tiniest blood vessels in the brain, a condition called cerebral small vessel disease, can lead to thinking and memory problems and stroke. These changes can also affect the blood-brain barrier, a layer of cells that protect the brain from toxins circulating in the blood. Now, a new study has found that people with cerebral small vessel disease who have blood-brain barrier leakage. Previous research has shown that disruption of the blood-brain barrier is increased in people with cerebral small vessel disease. People with cerebral small vessel disease also may have brain lesions called white matter hyperintensities. Such lesions are visible by MRI and believed to be signs of brain damage and a marker of the severity of the disease. For the study, researchers wanted to see if a leaky brain barrier was linked to degeneration of brain tissue, even before these brain lesions appeared. The researchers looked at normal brain tissue surrounding and close to the brain lesions, because they consider this tissue at risk. The study involved 43 people with cerebral small vessel disease, with an average age of 68. The researchers used MRI at the start of the study to measure the leakiness of the blood-brain barrier for each participant. They then used another brain imaging technique to measure the integrity of the tissue's microstructure surrounding brain lesions. This imaging technique was repeated 2 years later to see whether the brain tissue integrity has decreased. The researchers measured the relationship between blood-brain barrier leakage and changes in brain tissue. They found the higher the tissue volume with blood-brain barrier leakage at the start of the study, the greater the loss of brain tissue integrity was around brain lesions 2 years later. For every 10% increase in leakage volume at the start of the study, after 2 years the diffusivity of the brain tissue increased by 1.4%, representing a decrease in brain tissue integrity. They also found a similar relationship involving the leakage rate of the blood-brain barrier. A higher leakage rate at the start of the study resulted in more loss of tissue microstructure around the brain lesions. The research results support the theory that a compromised blood-brain barrier may play an early role in loss of brain tissue integrity as part of the cerebral small vessel disease process, even before brain lesions are visible on MRI. The change in integrity of brain tissue at risk, close to the brain lesions, could be a promising biomarker in future cerebral small vessel disease studies, examining possible prevention strategies and treatment options. A limitation of the study was that a short follow-up period may have underestimated the associations. Future studies should measure such changes in larger groups of people over longer periods of time. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video is strictly made for educational purposes only. I hope you enjoyed listening. Until next time, have a great day.